Hello students, today we're going to be talking about Stanovich Chapter 8. It focuses a lot on converging evidence. We've talked about a few of the points we're going to touch on today about theory and how you shift from one theory to the next theory in science. So we'll talk a lot about that today, both converging evidence in individual studies and how converging evidence from different fields and different kind of areas of a field can help us see more strength to a theory or that we need a paradigm shift. So, to begin with, let's review how we come to a paradigm shift, some of the things that we've talked about before, and with that, we'll mention the definitions of theory again. So, a theory is that big, broad set of ideas that can explain and predict the observations that we've made. So, at this point, we've already made tons of observations about how humans work, how human brains work, for example, how humans develop, and so those all are part of the theory, our broad idea, or explanation of how human development works, as an example. So with this, remember these theories need to be phrased or written or described in such a way that they could be falsifiable. So you need to be able to make, say, predictions from your theory and then be able to possibly falsify those, show that the theory did not come out to be true or did not make an accurate prediction. Then based on how many of those that it makes accurately or inaccurately, you've got to decide whether this theory is useful or not. So again, the theory is the really big, broad thing, versus the hypothesis is more specific in an individual study. So we get our hypotheses from what the theory suggests should happen. So theory suggests development happens this way, and based on that I might make a hypothesis, a single study hypothesis, that if I compare a child and an adolescent on this kind of skill, the adolescent might do better. So that's my specific hypothesis, that I hypothesize that the adolescent will do better than the child on whatever it is. But the theory describes that and more than that as well. So the theory is the big thing, hypothesis is more specific. The more hypotheses turn out to be credible and tested to be credible, the more strength that original theory has. So these are the main developmental theories in developmental psychology. Really, this is just a different model, a different visual representation of the theory. So it's the bioecological systems theory. So bio, like the biology of an individual, is important in this. Ecological, so the ecology, where they are, who is around them, the systems around them is how it's described. So microsystem, their family, people that they're kind of interacting with on basically a daily basis are part of your microsystem. The exosystem is like one step removed from that. So local politics probably affect your school and your family, but it's not necessarily having a direct impact. It's usually going through that pathway of affecting your family or affecting the school, then affecting you directly. So that is the big broad theory about how we change over time. I like this model of the theory because, again, we still have like community, society, culture as the environments that we're in, but it kind of gives us some more specific things to look at. So all these little arrows kind of remind us, oh, people are interacting with lots of different people. So an individual or a child, an adolescent, an adult is communicating with their school network. Maybe they're changing their school network, the school network is changing them, and an individual is made up of all these different pieces and more. So each of these is not, well, I guess you could say this one's a pretty exhaustive model, but I like the kind of focused attention that this model gives on a couple aspects of the theory. So again, that's the theory. Big, broad theory explaining how humans develop over time, being impacted and affected by the people around them and the systems around them. So that's the big theory. The specific hypothesis you might make, I might make a hypothesis that a child and an adolescent of these parents will both be affected by the parents and that they will affect the parents. And maybe I'd make a hypothesis like the young child that's very young is going to have a bigger effect on the parents than the adolescent who's like 15 or 16, because maybe that adolescent is already stepping out on their own a little more versus the young child might not have as big an effect on the parents. So basically a hypothesis is really specific, individual study wise. The theory is the big broad thing that will give you the ideas for what kind of studies or what kind of questions you need to ask or what kind of predictions are being made by this theory. So another example of a theory is Vygotsky's zone of proximal development model. So what a learner can do, what they can do with help, and then what they can't do yet. So if you can hold a bow for archery and shoot it a little bit, that might be what you can do. With some guidance, maybe you can actually hit a bullseye on the target. But what you can't do yet is probably be at a competitive level for a competition. But again, with more training, that circle of what you can do will expand. So that's the big broad theory. You might be able to make a hypothesis of how it would work for something specific like archery. So again, hypothesis is that more specific thing. All right, so hopefully you remember these things. I've mentioned them a few different times in the class, but a lot of it was way back at the very beginning of the class. So now that we're this far through the semester, I wanted to just kind of remind you of these things as we discuss how to get to paradigm shifts in the field. So a big piece of this is the quality as well as the quantity of data. So the quality of the data, is it confirming or disconfirming evidence? That's important, as well as the quantity, is it disconfirming or confirming? So if you have a really poor study and a really small sample that seems to suggest your theory didn't work out like you wanted to, is that going to be as convincing as a really big study, a really well done study, a really quality study that also shows that your theory didn't come out as well as you expected it to? You'll probably give more trust to the one with better quality, as well as if there's more quantity of lots of studies saying the same thing. So both the quality and the quantity is important as we're thinking about, is this theory useful? Is there things that this theory is not explaining well? That kind of thing. So remember also, scientists are focusing on the fringes of what's known. If there's something already known, we might replicate it maybe once or twice, or maybe replicate it in the process of doing something else. But once we figure things out, we usually go to the next thing that we don't understand. So that's why sometimes you'll see scientists have, for example, let's say I recently did a couple studies where it looks like there might be a much higher percentage of heroes in the general population. And I replicated this study one time, but it was with a pretty similar sample, not the same sample, different people. So with a total of, the quick math in my head, like about 900 people, about 13% of them were heroes, like had done something heroic at some time in their life. Now that's two studies, a little bit of replication there, but I want to now test this in another sample. So I went to a conference last weekend. I described this result. I said, this is really exciting. I want to see if this actually holds, if this is how prevalent heroism is. So next I'll go and check adolescents, middle adults, late adults, early adults. I will check different samples like college students. I'll check people in the workforce. I'll look and see if just general human population in the US probably is where I'll go next, that aren't doing the survey through the same method that I used before. I'll see, hey, does this turn out? So at my next conference, I might go and say, so I did think that maybe about 13% of people might be heroes, but really they're not. Right? So I'm trying to focus on things that we don't know yet. So because of that, I might be really excited about this result last weekend, but then next year, once I have some more data, I might be like, well, I was wrong. So it's not that that data was necessarily bad or wrong, but just that a bigger picture, a broader picture, a better sample, more representative, more quality and quantity of data might suggest that that original finding was not as solid as it looks right now, that it's a decent finding. Like two studies, almost the same percentage, that's pretty convincing. If it was a really big difference in percentage, I would be less assured of it with just two studies. But when it was almost the identical percentage between the two studies, that's kind of surprising to me. But I still do have to do more work to see if this holds out over different populations. So when we get a really well-supported hypothesis or a really well-supported theory, we're not going to retest those things. So another example that maybe is clearer to you, we know that blood circulates through arteries and veins. We know that. We don't have to retest that. We don't have to re-experiment. Like in the process of schooling, you might do that test just like as an exercise. But like the scientists that are doing work on how bodies work, they don't need to restudy that. It's clear enough that we don't need to retread over that. So it's pretty clear that the Earth orbits the sun. So some people might call these things laws, but as scientists, we don't actually use that term. I've heard it in like middle school and high school. I heard the term law is like higher than a theory, but no, for a scientist, theory is like the top level thing. So another thing about how this formatting on the slide got all messed up. Sorry about that. And there's a lot of text, but we'll go through these points one by one. So a big point of this chapter is misinformation. Misinformation and misunderstanding of people understanding psychology is kind of a big focus of the Stanovich book as a whole. But another thing that people don't understand is this process of moving from one theory to another theory. Oftentimes people think it just comes from one single study, one really dramatic example. You put the kite up and the lightning bolt hit it and now you know about lightning. So sometimes that might happen, but usually it's a slower process, more studies, more quantity of studies to get to that final answer, and using that theory for a while and realizing, oh, it doesn't really fit and answer all the questions in this area. Maybe we need to adjust the theory, and then they might adjust it for a while and then say, well, you know, with all these adjustments, it's still not working great for this other thing. So someone else comes and says, hey, this new theory explains everything the old one did and all the new information. That takes more than just one study. Usually takes multiple years to make these kinds of transitions. So when society thinks that a single really impressive experiment can change the whole understanding of the field, and that's what they're expecting, then when they see something like a study, like my recent study where I saw about 13% of people had done heroic actions, they might say, wow, and there's news reports, oh, this many percent of people have done heroic actions. And that might be the case, right? And that might change what we previously, because previously we thought heroes were much rarer than that. So the current theory is that being a hero, doing heroic things, is pretty rare. So this would be a big change to the theory. Like we'd probably adjust and be like, oh, okay, so more people are heroic. But that being said, people might take that and be like, oh, the whole field has changed. It's not rare anymore. Well, we're still not completely clear that that is for sure a change that we should make to our theories about heroes. So hopefully these examples help. A lot of people don't understand that this is how science is done, and that's why you're in this class, to learn how science is done, so that hopefully for some of you who are really interested in the science, you know how to do it. And these steps and discussions of how we change from one theory to another apply to all fields of science as well. So a crucial experiment is talked about in your book as the way that most people assume that theory changes. That one single experiment will solve the problems or help us understand it completely with one single study, one single experiment. What is the actual process is much different. We talked about the spontaneous generation theory and how for hundreds of years there were studies in trying to falsify the theory of spontaneous generation. It took hundreds of years because of the technology and measurement tools at the time to be able to really clearly falsify that things did not just generate based on other matter around them. So it's not a single experiment. To us, it looks like, oh, yep, this experiment showed that for sure the maggots did not spontaneously generate from the meat. But a problem is another study by another researcher, maybe fewer variables are controlled for, you get bacteria that grows even when the broth is boiled. So things happen that any given study, any given hypothesis might not come out. So you've got to think of the quality and the quantity together of the studies showing that support for old theory or support for new theory. So crucial experiments can happen, but they are very rare. So most often, multiple studies provide evidence towards parts of theories. So right now, we think, and even I still think, that heroes are probably pretty rare in the population. I have two individual studies right now, well, that are kind of published together as one study. So let's say one study that shows maybe heroes are a little more common than we expected. So now we have to do more studies. We have to get a broader sample. We get to get more quality data to be able to change that theory. We're not going to change it automatically. Even though I found the things, I'm excited about the findings, I'm not going around saying, hey, theory is different now. So when we think as a society that these crucial experiments are the way that science changes, we don't see a lot of the other studies done as valid or useful. It's like, well, in a study, you might be looking at some really specific pieces that you want to understand better about this theory. And some people will be like, well, that doesn't really change what we understand really dramatically. Well, a lot of studies don't change what we understand really dramatically. They're often little movements, so at a time. So society sees those many individual smaller studies as useless, which is really sad because that's what most of the studies actually are. And it's the building up of those bases of evidence across many, many studies that are very useful. That's one reason meta-analysis is, I think, a really useful thing to understand and know about, because it takes those individual studies and it puts all their information together and says, hey, based on these 40 studies or 20 studies or 200 studies, these are the results we see across the world, across the country, that kind of thing. So it's kind of an easier to digest and a really good method, a clear method about these things. So this idea that a single critical experiment or crucial experiment leaps forward the theory is also called the Great Leap Theory. So these advances are thought to happen in big leaps and bounds between the types of theories, but most of the times it's really little. It's small baby steps through, and small baby steps showing, oh, you know what, the theory doesn't do a great job here, still not a great job here, maybe we should adjust it this way. But then you might get to a point where you go, oh, you know what, this theory isn't explaining enough, like we need something new. Maybe this new theory will explain everything that the other one did and new stuff. So usually there's a lot of kind of fuzzy, blurry experiments where it's not so black and white, and those are those littler studies that are testing individual hypotheses. So with this, it has a lot to do with reliability and validity. So in your own notes, really quickly, just put down, how would reliability and validity have anything to do with this process? All right, feel free to pause if you, so that you have time to write that down. But reliability, again, is the consistency or replicability of a measure. So do you get the same kind of result every time you do this? If you're measuring weight, is your scale measuring the same weight every time? Assuming the weight shouldn't be changing, right? Validity, are you actually measuring the thing you think you're measuring? So, am I actually measuring heroism? Is my measure reliable? Is it going to be consistent? If those people told me about their experience later, would I still say, yes, that's heroic? Is it a valid measure? Am I actually getting at heroism, or am I getting at something like pro-social behavior? Because those are related but different things, right? So you need to make sure whatever measures you have are reliable and valid. And this is sometimes why any individual study might not find support for a theory. It also might be you do have valid measures, and so the evidence that's not supporting your theory is good, valid evidence. But it's a question you need to ask yourself, is your measure reliable? Is your measure valid? If we use a telescope to try and measure the distance between two seas on the moon, right? We use a telescope, we're looking through with our eyes, we chart it out or whatever based on what we're looking at, we can draw really, really well, and then we measure the distance between those. What if our lenses of our telescope are a little wobbly? And so it looks to us like they're closer or further than they actually are. So this goes down to the reliability and validity of tools. And that can have an impact on when we're testing these theories. So how do we actually reach paradigm shifts? I've mentioned it multiple times now. You need lots and lots of studies of people testing a theory and examining individual hypotheses. Any new theory that comes along, whether it's going from Newtonian physics to Einstein to quantum mechanics, moving from those things, the new theory has to explain everything the previous theory explained and all the new facts and observations that are being made that the old theory doesn't explain well. So you need to explain everything new and everything old, or at least more of the new stuff than the old theory did. And most of this chapter is how we do this in more detail. One way we do this is through converging or connecting data and studies together. So in order for any advancement, you need to explain the old and the new facts. If the advancement only accounts for the new facts but not the old ones, it's not going to be very useful. So falsifying a theory takes converging evidence. So converged or connected evidence. So, for example, evolution. We have a lot of converging evidence from lots of different fields of study suggesting that evolution is the best explanation for the development of organisms. So we have things like the genetic code. We can see evidence of evolution with it. We have experiments where we've seen even like fruit flies that live for like 24 hours. We've seen them evolve in actual experiments. That's hard to do with something with a lifespan as long as like years. But with things with very short lifespans, we can do that. We can look at the fossil record. We can see transitional forms in genetic code in the fossil records. We can see anatomical vestiges is basically like the parts of creatures that are left over by evolution that haven't been naturally selected away. So some snakes, for example, still have like leg bones sticking out, but they're not using them as legs. So we've got all this converging evidence to really strongly suggest that evolution is the best theory to describe how life developed on the planet. So this evidence comes from biology, from geology, from experiments, from the species distribution across the planet. So there's lots and lots of fields that give this evidence together. And that's what converging evidence is. You've got lots and lots of studies from lots and lots of areas, from many fields or multiple areas of fields that all point to the same kind of answer. So if we had a lot of other studies suggesting some other answer, or there's a bunch of evidence that didn't suggest that this explained everything, then we might need to think about adjusting the theory. So here's an example of the evolution of wings. So velociraptors, around the time velociraptors were, T-Rex probably had some feathers, but it was a little earlier and probably didn't have full wings. Then later on they had even bigger and bigger wings till we have the birds that are pretty common to us now, crows and chickens. So they are the great-great-great-great-great-great-grandchildren of the type of dinosaurs that T-Rex and Velociraptor were. So an example of a couple pieces of that converging evidence on this broad theory of evolution. Another example is positive upward spirals. So one thing that we've seen in many studies that weren't even necessarily looking for these spirals is that when you have a positive emotion, you tend to be more open to things in those moments that you're feeling that positive emotions. So you're a little more likely to be open to new relationships, to new types of thinking, to new things in the environment. And when you do that, you often build better relationships, you have more support from people, so you feel these positive emotions, open up more to people, build more resources for yourself, you have more health and survival and fulfillment because of those things, which in turn also produces more positive emotions. Let's say you have a hard day, you have a person that's now your friend that you can call and get some support, you get some more positive feelings, you become more open to what they're saying, you become closer with that person, so it's a big positive upward spiral. And we see this in things like depression research, we see this in happiness research, research on anxiety, we see this in animals as well, so like rats and other mammals. So there's a lot of converging evidence that these kind of positive upward spirals can happen. So many areas of human experience, we kind of broaden and build our capacities, and it opens us up and teaches us things and enables us to grow and develop. So positive steps can make the next steps easier, enhance our cognition, enhance the abilities that we already have. So if our memory is already this good, with a positive emotion it might be a little better. So it just makes things just that much a little better, a little easier, and makes it so it's more likely to have more positive things. So you can also have negative downward spirals too. But positive emotions or resilience can lead to other positive things. So emotion research, happiness research, developmental trajectories, longitudinal studies, therapy research. All of these things have shown us that these positive upward spirals are a pretty common experience for humans. So you could say that is the broad theory of one way positive emotions affect development. So it would still be under the broader theory of like the bioecological systems model, that positive upward spirals can exist under that. So all these kinds of converging evidence, they connect studies from different fields. This is the connectivity principle. When you see from so many different areas that these positive upward spirals happen, or if you see from so many different areas that evolution is the best explanation, it makes it more clear that your theory, the big broad theory of positive upward spirals or evolution, it makes it more clear that that is useful and accurate. So no single study is making all of these predictions about evolution. That would take a lot of money and it would be a really long paper. It would be probably longer than a couple books. For positive upward spirals, you could probably test the positive upward spirals piece in a single study. It would still be a pretty big study, like an entire journal length article or an entire journal's worth of articles. So usually there's like 5, maybe 10 articles, depends on the journal, that they put in their whole journal. And so about that much evidence for the entire system of the positive upward spirals. So these kind of relationships, like 1, 2, 3, and then 2, 3, 4, and then 3, 4, 5, these are all done in different individual studies put together. So converging the evidence. So scientists really like to look for these patterns on what does the evidence say? So the patterns of the findings, what are the patterns of the flaws? Do we need a better measure or method because we're getting the same kind of measurement flaws? What's being confirmed? What's being disconfirmed in our hypotheses? Leads us to decide which data supports and which data does not support our theory. So not every experiment or study is going to use the same exact variables or ways to measure something. Like you might measure heroism by asking people questions, have you done something heroic? You might use a scale, see how likely heroism is, something like that. But not every experiment is going to use both of them. Some might use one, some might use the other, right? Measurement is not always completely consistent between the times you do it. So you want a very reliable measure. Let's say you're measuring happiness. You want it to be a consistent measure. You would like if 2 researchers across the country were using this measurement of happiness, it still might not be super consistent based on other things that they might put in the survey as well. If they put a scale of depression right before their scale of happiness, it might affect their results, right? So patterns help confirm or disconfirm a theory. So we look at all this converging evidence and say, hey, does this theory have a lot of support or does it not? Do we need more evidence for and new explanations for what is happening here? So what methods should we use? Should we use correlations? Should we use quasi-experiments? Should we use true experiments? So think about it. If we're trying to look for a converging of evidence to support or disconfirm a theory, what kind of methods would be best? So take some minutes and write it down what you think would be best. All right, so feel free to pause and keep writing down. But really, the best methods to use are all the methods. So you might use interviews to understand something about the theory. You might use correlational studies for part of it and understand some of these relationships. You might use experiments to understand this a little better. And quasi-experiments if you can't do true experiments. Longitudinal studies to see how things change over time. Cross-sectional studies if you want to get quicker data about possible developmental change. So use a combination of all the methods. So any one single piece of evidence, any one experimental study, or any one longitudinal study, or any one interview study might give you some really quality data, but when you have the quantity, you could get really quality data from any one of those studies, But having 3 quality studies gives you a bigger quantity of information and data to show, yep, this theory does have support, or this is maybe an area where the theory is not explaining things super well. So multiple methods of collecting data, whatever is going to be the best, will help. So scientists try to come to consensuses about theory because of convergence between multiple individual studies flowing to the same kind of answers. So this is called the gradual synthesis model. So over time, you synthesize the results. You see, oh, the real prevalence of heroism is this, and we'll come to a gradual synthesis. The studies that have been done before, the studies I'm doing now, the studies I'm doing in the future, this will gradually synthesize and help us come to a clearer understanding of exactly this phenomenon in the world. Well, first probably focus on the US because it's easier sample for me to access right now, and then hopefully also the world. For that, I'll probably need to inspire people around the world to take up some of this research. There's still already researchers in, say, like Germany and Australia, but those are still pretty Western countries. So I would like to get some additional places around the world and additional colleagues to work with. So some more friends, and we can build relationships and do studies and learn more cool stuff. So we'll shift now from the changing of theories as a whole, so moving from one theory to another, to in any individual single study, we might want to use triangulation. So it is kind of similar in some ways to converging evidence, but it's converging evidence in a specific study. So with triangulation, you're essentially using multiple sources of data or multiple methods in one single study to make your finding more clear. So I could do something in a study where I try to get the prevalence of heroism, let's say 3 ways. One way, I ask people, hey, have you ever done something someone would consider heroic? Please describe it. I could do 2, a scale of intended heroic behavior. Hey, take the scale. The people who are higher on it are probably more likely to have done something heroic or likely to do something heroic in the future. And then a third one, ask like friends around them. Do you know of anything heroic this person's ever done? So now I have 3 types of data trying to get at, has this person done something heroic? That is a lot more clear. That's stronger evidence in that one study about their heroic behavior. Let's use another example. So let's say we wanted to measure happiness. One easy way to do that, we could use a scale of happiness. Say, hey, how often do you feel really happy? Scale from 1 to 10, like basically a survey about their own happiness. We can ask them that. A second way we can measure this too in the same study could be we ask their peers. So, hey friends, how happy would you say this person is on a day-to-day basis, like on a scale of 1 to 10? So now we have their number that they've given themselves and the number their friends have given them. Maybe we also ask, say, a parent or a boss or something like that. We could do that. So that's maybe 3 pieces of data where people are reporting about this person's happiness. But then also we get like a cheek swab to look at their hormone levels or something like that on a couple different days. And we do that every 2 days to get kind of a general average of about what their levels of like hormones related to happiness are. So now we have 4 types of data that are kind of getting at happiness. We can combine those into kind of one result that says, oh, all of these say you're at like the top of the scale for happiness, or you're at the bottom of the scale for happiness, or you're at the middle. So we have 4 pieces of data for our one number essentially. That's triangulating. So you're triangulating on happiness. So it really increases your credibility and validity of your findings when you use these multiple methods. Hopefully each of those individual methods are a valid method. And then if you're using multiple valid methods at the same time, you can be a lot more sure that your final result is valid. So combining those things in any given study really helps ensure that the fundamental problems of one measure are overcome. So asking people about their own happiness, they might overestimate or underestimate. Your friends might overestimate or underestimate, but hopefully those problems might cancel each other out. So it really helps to explore these complex human behaviors using a variety of methods in any given study. So that study is going to be, if you have 2 studies, one has 1 method of investigating happiness and the other has 3 methods of investigating happiness, the one with 3 is probably going to be a higher quality study. So obviously you can't always do that, but triangulation is really good. So how do we then come to that scientific consensus? A lot of times we use meta-analyses. We do a study of studies. A meta-analysis will determine what the true effect size is in the real world based on all the available studies. So in any of my given studies, I'll publish the effect size. So I found the difference between these groups when they did my program to go up this much, and I describe it in an effect size. Another researcher later on can say, okay, so this study did that program, then these other 4 studies also used that program. I can take all their effect sizes, combine them together mathematically, and see for all these studies what is the true strength of this program across all the different samples they tested. So it's a really good way that not any individual researcher, so it might be 4 different teams did these 4 different studies, and then another researcher might say, hey, I'm going to put all this together. It's a really good way to get a very, very clear picture of what's actually happening. So let's watch a couple videos. They're fairly short to get a better understanding of what is happening with a meta-analysis.
Hi, I'm Dr. Jessie Hawkins. I'm a clinical researcher and I'm the director of the Franklin. Hi, I'm Dr. Jessie Hawkins. I'm a clinical researcher and I'm the director of the Franklin School of Integrative Health Sciences. And we are going to kick off the Ask Dr. Hawkins series with one of the questions we actually get probably the most frequently, and that is, what is a meta-analysis? Now, we're asked this question pretty frequently because this is a type of study that our research team actually conducts pretty regularly. We conduct meta-analyses on aromatherapy, dietary supplements, behavioral interventions, and other health interventions. And the reason we conduct so many meta-analyses is because this is an important type of research that answers questions that cannot be answered in a randomized controlled trial. So a meta-analysis.
So quick point, a randomized controlled trial is basically a true experiment. You randomize people into your groups, you give one people the dietary supplement, one group gets the placebo, you see the results. That'd be a good evidence from an experiment, but this individual and their team takes multiple of those studies and combines it with a meta-analysis. So let's say one study shows that this dietary supplement is really good, but another study shows it didn't work. So what is the true result? Well, rather than just going based off of those 2 studies, a meta-analysis can combine those 2 as well as other studies that tested this same dietary supplement in experiments, combine their effect size results for more of a clear picture of what the true effect of this dietary supplement will be.
Exist to solve the problem of the ever-growing body of data that we create through other types of studies, epidemiological studies, and of course randomized controlled trials. So imagine yourself in this scenario where you've got maybe 20 randomized controlled trials in front of you, and some of them show that the
So again, randomized controlled trial, just a name for a type of experiment.
Intervention is effective. Some of them show that the controlled trials in front of you, and some of them show that the intervention is effective. Some of them show that it doesn't seem to be effective. Some of them there was progress, but it wasn't statistically significant, and you've got kind of this chaotic assortment of information in front of you. Without a meta-analysis, the options are to either pick and choose what we want, which then of course leads us to confirmation bias, or just keep researching. We can keep asking the same research question over and over, subject more and more participants to the same intervention instead of actually growing the body of knowledge and expanding beyond this research question. So that's why a meta-analysis was developed. A meta-analysis basically takes the existing research. It's a study of studies instead of a study of participants. And we take the existing research and we use it to extract data. So we take all the information. It doesn't matter if it was statistically significant in that study or not. We want to know the intervention, the outcome. We want to kind of aggregate all that data together and determine on our own what is the actual effect size of this intervention. And this gives us information that's actually going to be far more generalizable than what we could obtain in a single clinical trial. Because a clinical trial, of course, has limitations with generalizability that are not going to exist with a meta-analysis because we're using information probably from different populations. We're putting together quite a few different clinical trials. Another
way to put this would essentially be, I would do an experiment maybe with population here in Utah. Another researcher might do the same experiment with the population in Florida. Another researcher might do that same experiment with the population in New York, and another one in Montana, Nebraska, Indiana, California. So a bunch of people across the whole country. So any individual study might have some impacts of the culture, of the area, those kinds of things. A meta-analysis can say, okay, taking all of these participants, because it's all the studies of all those participants, and that makes it a lot more generalizable. Because while I might have only been able to generalize my one study in Utah to Utahns, the study or the meta-analysis can hopefully generalize to the entire areas that it was tested in. And with the examples that I gave, I'd be pretty confident in it applying to everybody in the US.
A really great benefit of a meta-analysis is that we can quantify the effect size. So we can be a lot more certain this is what this intervention is going to do, and this is how well it's going to do it. And that effect size is far more important to us, of course, than statistical significance. And finally, one of the greatest benefits of a meta-analysis is that it enables us to evaluate what we call moderator variables. And these are other variables. They can be demographic variables, they can be related to the intervention, they can be related to a lot of different factors that are going to influence or moderate how the intervention affects or produces the outcome. So
a good example of that would be, hey, does it matter from the studies? Does it matter if they were done in a place with a high population or like a more rural population? You could check that as a moderator in this kind of meta-analysis because you have the studies from Utah, Indiana, New York, California, Montana. I don't remember all the places. Florida. I don't remember all the places I said. But you could test that as one potential variable that you wouldn't have had access to with my one study of just people in Utah.
Is this intervention going to be more effective in women than in men, or in adults than children, or if combined with this other intervention? We can evaluate these things separately, analyze them, and then really provide more information again than we could get in a regular clinical trial. So
that is what a meta-analysis is, and that was a really good description. I don't remember if the next one has additional examples that I think are better. I think it's more of a visual example.
Trial dictates is.
So feel free if you're still a little confused on meta-analyses, but I think the examples given by both that researcher and myself, I think are pretty clear. But if you're still a little confused on meta-analysis, definitely watch this second video. I'm not going to watch it during class time right now, but hopefully that gets it really clear that a meta-analysis is a really good way to see what is the evidence converging on. Basically, we have these 20 different experiments. What do they all say together? So with that, you might be able to see a confidence interval. So basically what a confidence interval is, you might say the mean change or the average change of this group was they got a score 5 points higher than the control group, or they had a reduction in, I don't actually know how cancer cells are measured, but a reduction of this percent. So you can take that, but then also see, but based on everything we know from all these studies, you can get a really good, clear view of what we would actually expect. So what would the confidence interval be around this actual change? So basically, rather than just one specific number, it says, but we're about 95% confident based on all the studies we've done that the true difference, probably like 95% sure that it lies between these 2 points. And that's better than just our one single point from our one single study. So you can get confidence intervals in individual studies as well. But a meta-analysis confidence interval is you can be a lot more confident. There's a lot more consensus in that number or that range of numbers. So it's going to give us the effect size and confidence interval from many studies, not just one single experiment. So multiple studies and people testing a theory and examining these individual hypotheses, we slowly come to a paradigm shift. So we might see that, our theory is not explaining these new observations. Maybe we need to tweak the theory. So for the example of heroism, one way we might tweak it is we've always thought that heroes are rare. We can tweak the theory and say, well, they're not as rare as we thought, and then have some explanations about that. That might be a tweak of the theory. further in the future, we might realize, oh, you know what? Heroism isn't actually a separate thing to what we describe as pro-social behavior. So just as an example. And say, so really, this theory we were using to describe heroism is not the greatest way to describe it. This other way is better to describe all the ways we do pro-social behavior. So that would be an example of tweaking a theory versus a complete paradigm shift around the theory. So thinking about our developmental research series about how the systems and context affect an individual, and an individual can affect their context over time, that's a pretty big, broad theory. But even so, as well as it's explaining things now, it is possible that maybe with getting better measurement tools, we might start finding things that it just doesn't explain super well. So could there be paradigm shifts still? Absolutely, there definitely can. And this is the process to go from one theory, one paradigm, to another. All right, so that is all for Chapter 8. Hopefully the examples were clear. I know a lot of this was a little bit of repetition from like Chapters 1 and 2 of Stanovich, but hopefully you understand studies enough that we can build upon what you've learned to really understand how we do that kind of nitty-gritty process of moving from one theory to another theory in that paradigm shift.